Our next speaker is Dr. Yves Masson, who, he was the chair in the first session. So, Jim is actually kind of a mentor to me, so I'll be very happy to see his talk today about DNA double-stranded break repair and impact on cancer and the functional impacts of cancer mutations. So go ahead, Jim, whenever you want.
Okay, thank you. So everybody can see my screen right now? Yeah.
Yes.
Okay. So the presentation will be more into the cancer model aspect of this session. So I won't tell you too much about the regulation of DNA double-strand break repair, but I'll focus on the functional impact of cancer mutations. So the outline of the talk is here. I will first talk about a pipeline that we have developed to look at POLB2 functional tests, considering all the mutations that are coming out during sequencing. I'll tell you also about the interpretation of the effect of mutations, the functional analysis versus in silico prediction. And finally, I'll end up telling about our newest work in assessing the increasing number of VUS meaning a variant of unknown significance in DNA repair genes. So I'll go very quickly here. For many years, we've been interested into homologous combination, which is a faithful way of repairing double-strand breaks. And there are many steps in this process, but one key step is after DNA resection, where you create these single-strand tails here. The Rad51 recombinase finds homology into undamaged sister chromatid. And this is a process that is conserved during evolution. In bacteria, Rad51 has an homologue called REK, and REK can promote this reaction very well. And so basically, after DNA resection, REK or Rad51 will find the homology into sister chromatid, and this will prime DNA for synthesis. So over the years, we have found that, in fact, Rad51 is not as active as REK, and you need actually cofactors to stimulate Rad51 during strand invasion and D-loop formation. And such cofactors are the BRCA1, POLB2, and BRCA2 proteins. And interestingly, these proteins form a complex. So POLB2 is at the heart of the complex, so it binds on the on the N terminus through the cortical BRCA1, and through its C terminus, it binds to BRCA2. And it's interesting to see, and this will be important for later in the presentation, in the C terminus, you have a double D40 domain, which actually forms this barrel structure here to bind BRCA2. So over the years, I've seen POLB2 graduating from really like low risk breast cancer gene up to now people are considering POLB2 as a high risk breast cancer predisposing genes. And we know now that POLB2 mutations leads to eightfold increase of breast cancer. So this is quite clear now that POLB2 participates in cancer. So many years ago, we were wondering, what is this protein doing? So in the lab, we have a biochemical expertise. So we actually purified POLB2 to homogeneity. This took about 2 years. You can see here, or purified POLB2. And also a few years later, we actually purified BRCA2 to homogeneity. We have shown actually that we could reconstitute the D-loop formation structure here using purified DNA duplexes. So here we have resected 3' tail molecules, which are labeled at their extremities, and we know that when we add Rad51 in and also an homologous duplex, Rad51 is able to create these D-loop structures. And very strikingly, when we added POLB2 to this reaction, we saw up to a 20-fold induction in homologous recombination. So this told us that PALB2 is really important for homologous recombination. And actually, we could see that also in vivo or in cellulo. I have to be careful here. I always say in vivo instead of in cellulo. So we could see that if we knock down PALB2, there was also a decrease in Rad51 foci formation after using radiation. Like Ellen said, you can use PARP inhibitors to kill HR deficient cells. So basically, it was normal to see that in the PALB2 deficient cells, you have a high sensitivity to PARP inhibition. So all these studies really gave really nice, I would say, information on the biology of PALB2. But I sort of realized that all the assays that we developed over the years, the biochemical assays, the dilute formation, synthetic lethality with PARP inhibitors, it actually laid a very nice platform to start to look at what is the effect of PALB2 mutations of the US. So we actually worked with many people throughout the world, Heiko Vaneticum in Leiden, also Alvaro Montero in Florida, also Fergus Couch in Mayo, and hand in hand also with Jacques Simard at CHU Québec, Université Laval, to try to look at globally what is the functional effect of PALB2 missense mutations. So across 3 groups, here in Laval, in Mayo, and also in the Netherlands, we actually look at 134 different variations in PALB2 that actually span the length of PALB2. Of course, we could not cover all the possible mutations, but we covered several regions of PALB2, including the coil, which interacts with BRCA1, and also the double D40, which also interacts with the BRCA2 protein. So the first thing that we did is to try to establish a pipeline to monitor the functional effects of these different variants. And we reasoned that it would be probably wise to start with PARP inhibitor sensitivity, because at the end of the day, what you want to tell the patients is whether they are actually sensitive, well, will they respond well to PARP inhibition? So we started with a PARP inhibitor sensitivity test, and then we used several downstream assays to confirm that there was indeed an effect in the functions of PALB2. So we look at the recruitment of PALB2 to laser-induced double-strand breaks. I'll show that a bit later. We also look at Rad51 foci formation and also homologous recombination using a CRISPR-Cas assay that actually monitors very well homologous recombination in cellulo. So the pipeline is the overall goal of this pipeline is actually to be able to classify variants either as tolerated or deleterious. So we use an automatized cell survival assay to monitor olaparib sensitivity. To do this, we actually knock down PALB2, so you can see the endogenous PALB2, which is gone here, and we complemented with a YFP PALB2 construct, which is actually siRNA resistant. So within this construct, we engineered all the variants we wanted to assess, plate the cells into 96-well plate, then treat with olaparib, and then use automated nucleic counting to know what is the actual survival. So the first thing we learn is many of these variants, they don't do anything. So a vast majority here did not show any defect for olaparib treatment. However, we could detect 3 important variants, which are here. So the L35P, the T1030I, and the W1140G that were really sensitive to PARP inhibition. And you can see that here also with the survival curves. So obviously PALB2 is a DNA repair protein and is actually recruited to breaks. So you can see that here when you irradiate cells, we can see relocalization of PALB2 to the DNA breaks. So we confirm that these mutants were actually defective in the recruitment. So you can see here the typical recruitment of PALB2 wild type on the laser tracks. And this is the L35P, which is actually in the coil region of PALB2. So this mutant doesn't interact with BRCA1. It's actually a known pathogenic variant for PALB2, probably the only one. And also for the two other variants, we could also see a defect after recruitment to laser tracks. So a challenge that we face is that the classical functional assays that we had were really low throughput for many variants. Because when you deal with so many variants, you want to be able to assess very quickly, for instance, a number of foci for RAD51. So we implemented a high throughput DNA damage foci analysis platform on a Cell Discoverer 7 system. And here what we do actually is that we can specifically identify SG2 cells using a cyclin A marker. Okay, you can see that here. And then we can look specifically at RAD51 foci formation, and these are in red here. And we can count the number of foci which are actually produced. So this actually speeds up a lot our research, because now we can do that on many variants, and this will be important for the last part of the talk. So obviously we could see that the L35P here variant was defective in RAD51 foci formation, and also the two other variants were also had defects. As I told you, PALB2 is a DNA repair protein, so we expect that it would be a nuclear protein. We can see that here, but something that we observe in these two variants, the 1030I and the 1140G, is that some of the PALB2 was actually accumulating in the cytoplasm. So this was very interesting, because a few years ago, we have shown that if you actually truncate the double D40 domain of PALB2, you reveal a nuclear export sequence, as shown here. So if you use a truncated mutation, which is actually found in cancer, you reveal this NES sequence, and PALB2 can accumulate in the cytoplasm. So it was very interesting to us to see that a single point mutation can actually recapitulate this cytoplasmic accumulation model. This also tells you that, this type of research looks tedious, looking at all these variants, but you can actually discover some mechanisms that are linked to the different variants. We also use this CRISPR-Cas HDR assay that I mentioned previously. And in this case, what we do is we use Cas9 that will cleave in a lamin A locus. And following homologous recombination, you will insert mRuby reporter in the locus, so you can actually score mRuby positive cells as a surrogate for the levels of homologous recombination. Again, we could see that these three variants were defective in CRISPR-Cas induced HDR. So far with this study, we have shown that the PALB2 variants are actually located in two distinct regions. And the first region is really the coil of PALB2, which interacts with BRCA1, and also the double D40 domain, which interacts with BRCA2, but also RAD51C and RAD51. Another thing that we actually learned from this is that the predictive tools, the in silico predictive tools for the variants to see whether a specific variant would be benign, intermediate, or damaging, they're not really good for PALB2. So in the case of L35P here, although all our assays have shown that this is a damaging mutation, the Revel program here predicted that it would be benign. And if you take this benign, known to be benign mutation here, you can see that we don't have any effect in our assays, but the polyphen-2 and align GVGD programs actually predict this variant to be damaging. So this tells us that at least for PALB2, you really need functional assays to monitor what is the effect of these mutations. So this is the late summary of what I said, and I'm just going to jump on the ongoing experiments that are going on in the lab. And actually, well, we monitor 134 variants, but now we can see with all the sequencing from patients, and that there are more and more variants to be analyzed. So, for instance, if you look here in 2013, if you look at PALB2, there was only a few variants that were classified for PALB2. And in 2019, we're at more than 1,000 variants to analyze. So we started some saturation mutagenesis approaches in collaboration with Leah Staruta and Doug Fuller. And the idea here is to measure the effect of thousands of missense mutations on the protein function or stability. So basically here you have the amino acid sequence of PALB2. So we take every amino acid and we actually mutate it. So you have the 20 possibilities of amino acid at each position. So basically with this, you create a pool of clone containing all possible variations. And then you can test by functional assays whether the variants are sensitive to a particular drug, for instance, PARP inhibition. There are many advantages to this. First, all the information is self-contained. You have also all the information in one experiment, so that's quite interesting. So Larissa Milano, the lab, started to do the PALB2 site saturation variant library preparation. So we actually generated a PALB2 library where we have all the possible mutations. So PALB2 is 1186 amino acid. If you multiply by 20 variation per amino acid, we're nearly at 24,000 possible variations. So each variation here is linked to a barcode, so you don't have to sequence everything all the time. After a screen, you can only sequence the barcode. And these constructs are all inserted in a safe harbor locus of either knockdown PALB2 cells or knockout cells. Then the idea is really to treat with PARP inhibition and look at which variants are neutral or deleterious. I think I have 2 slides left. So Gemma here, Montalban, who is working in co-direction with Jacques Smar and myself, is doing exactly the same thing for RAD51C. In our case, RAD51C is a much smaller protein, so we have 7,500 variations, and it seems that RAD51C is a good ovarian predisposing gene, so I think this will be interesting to see which variant respond to PARP inhibitor treatment. So the idea is to create a database, an atlas of all possible variants in these 2 genes. So in 10 years, 20 years, somebody will come and say, hey, I have this methionine that is actually mutated in glycine. Is it deleterious for PALB2 function? And will I be able to take PARP inhibitors? So our idea is really to create this encyclopedia of all possible variants for these two genes. So I think I'll end there. So I want to acknowledge, obviously, the work of Amélie Rodrigue and Yann Coulombe for the first part of the talk, and also Larissa Milano and Gemma Moltaban for the last part of the talk. With the pandemic, I was hoping to have this data. Well, we were all hoping to have this data, but I guess this will be for a future conference.